Okay, I think we're ready to begin the next last part of our session. And it is a great honor for me to introduce Chris Sander, who is at Harvard Medical School in Boston and has been involved in one way or the other with the PDB related activities for many years. He's going to give us a talk on protein folding computed from evolutionary information. Chris.
Right. Well, thanks very much for all the speakers that talked today already, and Stephen and Helen and David for organizing this. I want to tell you a story that is somewhat colored by the work that we've done in the past, going way back, as you see in a minute, using the PDB early on, but also extrapolating into the near future in terms of all the interesting developments that the community has made and is making. All right. So the two stories I have is both related to evolutionary information, one from natural evolution on the left and the other one for experimental evolution on the right. The one on the right, in fact, relates somewhat to Francis Arnold's talk. And making the point that in 2011, we were the first to routinely compute correct folds from sequence information in 3D. And then recently developed the fourth experimental method for protein structure determination, following crystallography, NMR, and cryo-EM. And so the problem for the first one, the computational one, as you know, goes from amino acid sequences through a computational method to 3D structures, and that had been unsolved for quite some time. We approached it by using not mega computers, but using the lens of evolution, which in some sense was a new approach to an old problem because everybody was trying to do molecular dynamics to fold proteins. And evolutionary information, as you know, is extremely rich. It's in the form of multiple sequence alignments and other ways of analyzing sequences. And I've knew this early on in the work that I did at the Weizmann Institute, actually, that this was going to be really an important source of information. It took a while to get there, but the key point is that you have that we distilled from evolutionary information is the fact that when you have a contact on the protein here on the left, you get correlations and the multiple sequence alignment between two positions. The inverse, however, is very hard because you have many different confounding effects, such as transitive effects. So the way we solved this in 2010 is to take evolutionary multiple sequence alignments, and sequence alignment methods are extremely important in this, added a statistical model of some sophistication, but simple in the end conceptually, that basically says there are single residue terms and paired terms. And statistical physics tells us a lot about how to analyze such things in spin systems. And this is a spin system with not just 2, but 20 different states. And from that, you derive contact maps by solving the parameters in this mathematical problem, this JIJ, which are the couplings between two residues, provided that you have enough information on the left. And once you have enough correctly predicted contact, which are the red dots here on the background of the crystal structure contact, which are the gray dots, if you have enough of those coverage, you can actually put them in the molecular dynamics of various types, CNS, Rosetta, Open etc., and fold up the protein and get a 3D all atom model. And so this turned out to be a breakthrough in terms of using evolutionary information to get the correct folds of structures. And I'll add a little bit of personal history, partly because it relates to the PDB. I first tried contact prediction in '79 at the Weizmann Institute, working with Schneider-Lifson. And you see here beta strands and we do statistical models for contacts between residues, which are either single or cooperative. And then I was sliding the beta strands as a postdoc left and right to see whether I would get a different score when there was a different folding. That was the first attempt of cooperative contact prediction, at least in our hands. And at the time, I was using 30 PDB structures that I got on the tape from the PDB. So that was good. Without that, as many people said, I wouldn't really do it. The slight anecdote is that the PDB has come a long way. When I sent the tape, we wrote a program called DSSP, which some of you have know about, where we just analyzed helices by pattern recognition and strands. And then I wanted to distribute this with the PDB. And the message came back that, no, we can't do that because your program has lowercase letters and we can only do uppercase letters. And so computation has come a long way, and the PDB has come a long way. It's now a completely modernized operation over the last several decades, actually. And Helen Berman was a major driver in getting that done. And but then after this attempt in the mid-90s, we attempted to do protein contact prediction using multiple sequence alignment, because I'd learned by interacting with molecular biologists that they were able to do mutations in proteins. And learning from Manfred Eigen, I was just dreaming of being able to do evolutionary experiments, which I'll tell you about in a minute. So we took multiple sequence alignment and predicted contact maps structured by the bottom, and not bad actually at the time in 1994. These black dots are the correctly predicted contact, and the open circles are incorrect. Using two different methods, actually, either with or without phylogeny. And so that led to the idea that this is possible. We weren't the only one, though, at the time. Erin Neher in 94 and Willie Taylor also published papers having the similar idea of using correlated mutations for contact prediction, but they more or less concluded that it wasn't possible or probably not possible, but we were much more optimistic in the papers we wrote. And since then, and of course, this is not the only intellectual thread that went into it. Crystallographers had observed correlations of mutations in structures like Aaron Klug et al. But there were hundreds of papers following this, with people trying to do the same thing and increasingly better, but not really solving it until 2010, when we had 14 years later, many more sequences and a better mathematical model. And the mathematical model, in fact, was the maximum model that leads to this formulation of a Potts model that allows one to then solve the parameters, and then several mathematical ways of actually getting to the parameters from the multiple sequence alignment, and now routine. And so how good is that? Well, so in the first few years, you can see here the numbers. We did quite a number of large proteins all the way up to 600 amino acids with this kind of accuracy, made it available to the community. Others picked this up. David Jones immediately, David Baker about a year or two later. We talked to them at the end of 2011, and of course later on DeepMind picked it up. Now, how good is this method? Well, it's one thing you're doing it on known structure, the other one's to actually go either to CASP or compete, which is quite complicated and sometimes unclear process. We published predictions of membrane proteins. Oops, where around here, I got to go back one. Predictions of membrane, transmembrane proteins, at the time still difficult experimentally in 2012. And of the ones that were solved, about 6 or 7 or 8 now actually were correct at this level of accuracy, correct fold for sure, and reasonable coordinate accuracy. And so that was encouragement that this method was actually working. And since then, Deborah Marks in particular and her group and with my collaboration solved a number of other problems, including RNA folding, ribosomes, RNase P. We then looked at complexes. David Baker's group had a very nice papers, in fact, came out a few months before ours, using complexes, just evolutionary couplings between proteins to get protein complexes, interactions that go through a ligand indirectly, relating to how distance residues can also be correlated, and information transmission, and then also alternative conformations in proteins, where the overall conservation patterns of evolutionary couplings reflect both conformations. You have to deconvolve which one is which, and that still needs a much better algorithm, actually. Also, actually flexible proteins, which people call disorder proteins, or misnomer intrinsically disorder proteins, that actually they're flexible and they attain a structure when they interact with something else. And so since then, this was adopted by many groups, immediately by David Jones. Onichik then followed proteins. Then the following year, David Baker's group with Ovchinnikov and so on, Zhang, and then Xu, then using some machine learning methods to do contact prediction. And then, of course, later on, over several year effort, but first made public, John Jumper and others at DeepMind. So one interesting example is how to use these evolutionary couplings derived from multiple sequence alignment with other method, either NMR or, in fact, crystallography, molecular replacement this is already mentioned. Wayne mentioned it. So here's a nice example. The first one I think that was ever done, actually, in collaboration with Andrew Kruse, where we use EV fold predicted models to actually avoid phasing and derive data sets to actually solve that protein structure that was actually difficult to solve. And so this is proper crystallography. The solutions by doing the molecular replacement search, actually you see here in the unit cell the various solutions. The best solutions cluster in one region, and that cluster was then good enough to actually, in fact, to actually then solve the phase problem for rod A, an interesting transmembrane protein in bacteria. So you know, Randy Reed's done work on that, and others have. So this is actually one where predicted structures can actually be quite huge. Wayne mentioned that. Won't completely replace it, of course. And then since then, also others picked it up. Then sometime later, the AlphaFold team, Jonathan Jumper, coming out after several years of work, actually, with a very large group of people in 2018, and again in 2020, published this now, 2021. And David Baker's group, who of course had done amazing work on just folding proteins from molecular dynamics, of course, the whole protein design thing, but on also adapting machine learning methods to do something that built on what we had done, but does a better job in actually refining the structures. Just to illustrate that, this is just the workflow from the paper of the AlphaFold 2. They do have a pair representation in it, which is exactly what we pioneered in 2010, and they use multiple sequence alignment to extract the information. They do an iteration on that, which actually by putting attention weights on rows and columns of the multiple sequence alignment, that's actually an improvement. But then most importantly, they do a much better job at getting the side chains and coordinates right to higher accuracy. I'll make a comment on that in the end, how good that actually is right now. And the news items at the time, well, the 2 time points were that, you know, tongue in cheek, in 2011, this was the problem that had been unsolved for 40 years, and the breakthrough using evolutionary couplings and getting folds right for many proteins. And then again, 10 years later, a problem that had been unsolved for 50 years. And that now has been solved by the new method that uses machine learning and do a much better job at improving the accuracy of atomic positions in a way that will be broadly useful, however, not necessarily for drug design. And there's still quite a lot of, so there's a conceptual advance, roughly speaking, and some technical mathematical one in 2011 and more technical advance in 2021. So that's the first part of it, which is a trajectory in my mind with a bit of persistence. If there are any graduate students or postdocs listening, persistent sometimes counted. You know, I started this in year 7 of or year 8 of the PDB, and believe it or not, 30 years later, I managed to solve the basic problem, and now it's even getting much better. So the second part of it is what I call tongue-in-cheek. Not tongue-in-cheek, actually, it is the 4th method, experimental method, distinct experimental method of solving protein structures experimentally. And we did this. My dream was actually instigated by Manfred Eigen, with whom I like a long time ago, and Schneerlifson and so on, to do evolutionary experiments. I was able to do it. And what we did basically, and this is not directed, it's like directed evolution, but actually it's directed evolution, but staying in space. What we took is antibiotic resistance proteins, not screening, but selection, actually, quoting Francis, using beta-lactamase, for example, and this is AC6. And we just generated by a random process that was quite elaborate, 20 rounds of mutagenesis and then selection in bacteria, and getting increasing number of sequence all the time, selecting the bacteria which actually resisted a particular concentration. It was technically quite tricky, particular concentration of antibiotics, so that in the end we had for the larger protein 300,000, and for the smaller protein 1 million sequences generated in the laboratory with evolution, like directed evolution, but basically selecting the bacteria on the growth advantage. This can be generalized if you have a screening, a selection method, or the selection method at higher expense would have to be replaced by screening methodology. So from these large number of sequences, and then now the complete analogy using the same computational methods as we did for natural sequences, you get a contact map, and the red ones are the correct ones, and the gray ones was the known crystal structure. We did this on known proteins, but hiding the information, and we get computed 3D structures that are, are not as good as a 1.5 or 2 angstrom resolution crystal structure, but are getting there. They're correct fold with many details correct at the level of about 2 to 3 angstrom or so, 3.5 angstrom variation in some regions. But this is a demonstration project to show that without any crystallization, without any purification of a protein, for either cryo-EM or for NMR, just doing a genetics experiment, you can actually get the 3D structure. Now, will this replace crystallography? No way, but it's an interesting variant of how genetics and structure determination can be selected, just exploiting the principles of evolution in a very, very straightforward way. But you got to do it. Related to that, just very briefly, there's another one less expensive experiments. If you can actually measure, rather than select on growth, but you can actually measure the phenotypic consequence of a mutation at a real number level, an actual real number, not just a binary number, then you can do epistasis calculation for the single mutation, the double mutation, and pairs of mutations. It actually turns out you can get away with doing only maybe 10% of all pairs, and you get a reasonable accuracy by doing epistasis calculation and then filtering for the most epistatic pairs and predict those as predicted contacts as shown in this map, again, compared against the background of the known structure. And you can actually get demonstrated only here for a small protein, a few small proteins, you can actually get a crystal structure, a 3D structure from a genetics experiment that measures fitness and does epistasis calculation. So either by selection or by screening, these are 2 genetics methods to get protein 3D structures. So I call this the 4th experimental method. And people say, well, come on, but this is all computation. Experimental methods, previous experimental methods, and the ones which, of course, the most powerful, crystallography, NMR, and cryo-EM, need data. Yes, everybody needs data, but they also need heavy computation. I mean, crystallography was solved because people, in part because people are clever enough to solve the phase problem, et cetera, et cetera. And of course, Wayne is a grandmaster at this, and others that are listening here. And NMR needs heavy computation to actually do distance geometry to get correct structures. And cryo-EM, like nobody's business, needs heavy calculation. So this also needs heavy calculation, but the data is a different type of data. It's gene sequences. So that is something that we have shown. Now, jumping forward, oops, where are we? Yeah, jumping forward. Just one last comment on the recent AlphaFold and the Rosetta Fold measure. So wait a minute, this is not quite. I think I need one slide here. I think I'm missing one slide. Okay, there's both AlphaFold and Rosetta Fold have just come out recently with great accuracy, but you got to be careful. So they looked at, in addition to what they reported in the paper, they looked at 3,000 newly solved protein chains in the camera one, all the ones that came in the database, without looking at them. So blind test. But many of these are just similar structures or structure that can be easily modeled by template, what's called template model building. So this accuracy here in this graph, where 60% are under 2 angstroms, most of those are simple biomedicine model building. And so it's likely lower accuracy with proteins which are non-homologous to others or completely new folds. So this dividing line here would shift to the left if you only restrict yourself to the ones that are not already easily accessible to model building biohomology. And so in that sense, certainly the protein structure prediction problem isn't solved, even in spite of the great advances, but there's a lot of territory still to be covered. And of course, both crystallography and cryo-EM and NMR will continue to solve structures in a way that is absolutely unprecedented, especially cryo-EM recently, which I think is totally exciting. So some of the challenges then are that are for the computational part is folding from single sequence information. Everybody's using multiple sequence alignment, like I showed, or to actually calculate or follow the ensemble of folding pathway. Is that important? Well, it might be important, especially for the importance of remote interactions in the final structure that might actually play a role in the folding pathway. or alternative conformations, which are extremely important functionally. And the whole question of functional interactions, which the evolutionary couplings give you to some extent, but you want the functional interactions between proteins and substrates, et cetera, et cetera. So these are all challenges going forward, both for computational and experimental methods. So my acknowledgements for this go to a lot of collaborators, people with whom collaborated in statistical physics in Torino, Deborah Marks in particular, who after the initial opening of this just has done amazing work and solved many of the problems. And we're going to collaborate going forward on these challenges. So thank you very much.
Thank you, Chris. That was a really great talk. We can welcome some questions from the floor. Wayne? Wayne? You're muted. You're muted. Okay. Oh, now you're muted again.
Now his name is Anne.
No, that's his wonderful wife.
I know. He's great too. Wayne is one of my heroes, believe it or not. I mean, it's just such amazing work.
Wayne, you keep getting muted. Okay, try. No, it's mute. We can't hear you. Sorry.
Wayne, perhaps you could put your question into the chat. We are not hearing you even when you're unmuted. Sorry.
It may have something to do with the earbuds.
Yeah, no, unmute. Yeah, now try again.
No.
Now it's muted again.
No, we're just not hearing you. I'm sorry.
Okay.
Maybe while Wayne, can you try one more time? Can you? Okay, well, while Wayne is trying to sort that out, Chris, I obviously your focus was on proteins, but I saw in one of your slides that you talked about RNA. Are you spending time on that now or not?
Yeah, we are. So I mean, we followed the ribozymes, for example, which was just absolutely astounding because the evolutionary couplings on the ribosome, the strongest ones, are actually in the position in the ribosome where the ribosome goes through conformational changes as a result of something binding. It's just astounding. There are two residues have to be sort of, if they change, the other one has to change as well, and or the RNA protein complex like RNase P. So we have a project, but we haven't solved it. You know, I'd love to find ribozymes in the human genome and mammalian genomes. People have tried that, but not found any. So I think if we actually use the constraints as part of the sequence search, then we might be able to find them, but it's not done. That's one particular interest. And the other one are viral RNA. So we've worked on the SARS-CoV-2 virus, and the preliminary data not yet published, where we actually looked at the interactions at the level of RNA that affect both the replication process as well as the interaction with proteins that bind the RNA in coronaviruses, in particular the interaction between N and C terminus. So those are things ongoing on the RNA space.
Much needed. Wayne, can you try to ask a question again? Let's see if it works. No, it's not working. Perhaps you could type it into the chat, and then when we have the overall discussion, we can maybe between now and then we'll sort this out. Okay. Are there any other questions that anyone would like to ask Chris? Okay, so except for the fact that I would love to hear what Wayne has to say, and we can't, I think we should thank Chris again for a really stimulating talk. And I have to
Yeah.
say, Chris, that as you were speaking, I had all these questions, and every question I had, you answered. So
I just make one sentence comment, which, in terms of the both the alpha fold and the Rosetta fold are definitely progress, because it's now in the news, et cetera, et cetera. Others and we are actually going through the data with some care to see actually what's the dividing line between the easy ones, which are remote homology buildings, and the hard ones, which are unknown. And so we'll have to look at that data more carefully to see actually, and then try it out. And running this on own examples, and then see, where can we actually help take this even to a next step, and also how can we use it broadly in the way that people are suggesting. But this isn't quite, it's very well advertised, and we have to see actually what it really means.
yeah, that's really, well, hopefully we'll discuss this some more at the end.